Okay guys, hello, this is Tam and Tom and we're going to talk to you about enzymes. So, can you grab a bit of paper and I want you to pause the video and answer these questions. Okay, so if you could pause the video now. Okay, and we're going to whiz through these questions, sort of they're, I guess, GCSE review questions, so they are quite important. So first one, what does an enzyme do, Tom?
Okay, well, enzymes catalyse reactions, which means they speed up the rate at which the reactions go at. And without enzymes, the rates of reaction would be so slow that there wouldn't be any life. So quite important.
Be a boring world, really. Yeah. So there we go. Catalyse reactions, speed up rate of reaction, digestive and metabolic reactions. Again, the two different types. Okay, so again, Tom, where do you find enzymes?
Well, everywhere. I mean, probably from GCSE you concentrated mostly on digestive enzymes, so enzymes that were secreted into the gut to break down the large food molecules. But actually, they're only a very small part of the enzymes in your body. Inside your cells there are literally tens of thousands of enzymes, more than that probably.
Yeah.
Hundreds of thousands of enzymes, all catalysing different reactions. So here we've got, that's a lovely picture of a cell that's been stained with fluorescence.
One of my students actually, he told me that he did some research with cancer cells in the summer.
Wow.
And he's got some photos like this. I've asked him to send them to me, but they're awesome. He's dyed all the proteins and stuff like that.
Yeah. You can see the nucleus blue and the cytoskeleton's coming out green, I think.
Yeah.
Very nice. And there's some your old digestive enzymes, and you can also have enzymes in washing powders, which have a biological origin to help wash your clothes whiter.
Yeah, absolutely. And people can be allergic to them as well, I think. Okay.
Me?
Are you allergic?
Yeah, I can't use biological washing powders.
You're a sensitive soul there, Tom.
I am.
Okay, so you should remember some of you should remember from GCSE, perhaps not everyone, but enzymes are made of amino acids, okay? So they are globular proteins. So you can see here on the picture, top left, obviously that's an enzyme, and we're going to talk about the lock and key model in a second. Bottom left, that's a diagram of an amino acid, okay? So again, you guys will have done proteins, so you should be able to draw out an amino acid. And the residual group, again, you change that depending on what type of amino acid it was. Okay, so can you name any enzymes? Tom, tell us some enzymes.
Well, there are loads of enzymes, as we mentioned already, and you can always recognise it's an enzyme because in 99% of the cases they end in A's. So lipase, protease, those are both digestive enzymes involved in breaking down large molecules to small ones. DNA polymerase is an enzyme which builds up monomers into larger polymers, so that's sort of going the opposite way, making small molecules into big ones. And ATPase is a really important enzyme which
helps you live, really.
Yeah, so that phosphorylates ADP to produce ATP, which is the energy currency in the cell. So you can have, sometimes they've got really long names like ribulose bisphosphate carboxylase oxygenase, which is also known as Ribisco, and so just look at the end. If it's A's, it's an enzyme.
And ATPase actually you find in mitochondria, as you guys are probably aware from that. Okay, so if we then look a little bit at how enzymes catalyse reactions, it's sometimes quite nice to imagine that when a reaction's taking place, you have to put a bit of energy in first of all in order to get the reaction going. Okay, so it's rather like this little guy pushing a boulder up the hill. He has to put in quite a lot of energy to start off with to get it to the top of the hill, and then the reaction can take place, or the boulder can roll down the hill. Okay, and that kind of translates quite nicely onto like kind of energy graphs to sort of represent this. So this is just like a little pictorial representation, I guess. So you can see initially this is our first reaction, and we've got a fairly large activation energy. So again, the amount of energy that you put in. So if, for example, it's lighting something like a fire, you have to put in a bit of energy first of all to get that heat to get the fire going. But if we add an enzyme, what actually happens is that activation energy is reduced an awful lot, okay? And that helps with a lot of things. So, Tom, why is it important that we reduce that activation energy?
Well, as you were saying, if you've got a very large activation energy, then it's very unlikely for the reaction to sort of happen spontaneously, if you like. So that activation energy is the energy you need usually to break a few bonds in the substrates or the reactants to get them to turn into the products. Whereas what an enzyme does is it usually grabs the substrate or substrates and holds them in such a way as to make that bond breaking more likely, and therefore have lower energy to trigger those very important activating reactions, and then so the reactions actually happen at sort of cell temperature. In chemistry you might have to get a Bunsen burner under something to get it to go, whereas obviously that wouldn't work if you were in a cell, because you'd burn everything up. So you have to go through a different route, and that's what the enzymes allow.
Yeah, absolutely. Okay.
I like those guys.
Yeah, I like those guys. Okay, so if we were to then have a little think about what exactly an enzyme does, okay? So can you guys please pause the video and have a go at this question, okay? Okay, so lock and key theory, you should have something that looks a bit like this. We start off with our substrate, okay, so our substrate in this case is sucrose, and sucrase, because it's got A's on the end, is the enzyme. So we're going to collide those two molecules, or they're going to collide through movement, random movement, and we create something called the enzyme substrate complex, okay? And then that's going to react, and we still have our sucrase in exactly the same structure. The enzyme hasn't changed at all, but the substrate has now been converted into a product. And as you guys know from learning about sort of your carbohydrates and stuff like that, sucrose is a disaccharide, and it's made up of glucose and fructose, the monosaccharides.
Cool. Can I just say, your pictures don't have to look exactly like that. The important thing is that the substrate is a complementary shaped fit to the active site. So I usually do them as like a little slice of pie out of a big cake. But, you can do them any way you like.
Yeah, this kind of looks like a... no, actually.
Yeah, that little bit.
It's an odd-shaped cake.
A little triangle-y bit.
Yeah, I think it looks like a mouse, doesn't it? Sort of, anyway. Okay, so there's another theory that's not the lock and key theory. It's slightly different, and it's called the induced fit theory. So, Tom, can you tell us?
Well, this is probably more like what's going on. So in this case, you can see the substrate and the active site are very similar shapes, but not exactly the same. And when the substrate binds with the enzyme to form the enzyme substrate complex, it causes a slight conformational change in the shape of the enzyme, which sort of snuggles round the substrate and holds it tighter, maybe putting strain on some of those bonds that need to be broken to get it to turn into the products. And so I like to think of it as like a pillow. The enzyme's like a pillow, and you sort of puff it up, and then you put your head on it, and your head's the substrate, and the pillow sort of folds itself around the side of your face, inducing a complementary shaped fit to the shape of the substrate.
Good. So then you get this really close fitting sort of active site on the enzyme and the substrate as well. Yeah, most pillows should do that. Not always.
No, it depends how old your pillow is, I think.
I had a really bad one in Barcelona. It was like a rock. It was awful, yeah.
Nothing worse than a
bad memory, yeah. Anyway, so we're going to have a quick look then at inhibitors. So a couple of questions here that I'd like you to, again, pause the video and have a go at maybe looking these up on Google or something. Okay, so pause it now.
Or in your textbook.
Yeah, or in your textbook. Okay, so let's talk then about these questions. So, Tom, what is an enzyme inhibitor?
So, like it says really, if you have an inhibitor, it stops the enzyme catalyzing the reaction or slows down the catalysis. So it reduces the rate of reaction in some way or other. And there are two types of inhibitor you need to know about.
Yeah, absolutely. So basically how inhibitors work is, if we have a little look at these two different examples, as Tom said, we've got one called a competitive inhibitor, which is showing on the screen right now. Okay, see that big blue blob? that is the inhibitor, and it's sitting in the active site, which is the site where the substrate would normally bind. So it's competing directly with the substrate for that active site, okay? And it prevents the substrate from binding in there, and so it stops the reaction. Okay, not very good. This is our other inhibitor. So Tom, what's going on here?
So this is a non-competitive inhibitor because it's not competing with the substrate to get into the active site. It actually binds to some other part of the enzyme, which is sometimes known as the allosteric binding site. And when it binds to that, it changes the enzyme shape such that the enzyme's active site is deformed, and so the substrate will no longer fit into it, and so it won't catalyse reactions.
Yeah, so we can see absolutely that the active site on the enzyme, now it's kind of the sides have folded in a bit and the substrate's not able to fit.
Yeah.
Okay, so it's not competing directly for the active site. Brilliant. Okay, so now's, I don't know, some of you might know this, but basically some people are able to recognise and smell cyanide, and it's thought that sort of back in like olden days or whatever, you'd have like specialised people who'd go into morgues and whatever and sniff out victims who'd been killed by cyanide. So you can see this guy at the back. Well, he's thinking, can I smell cyanide? I'm not sure. So why is this relevant, Tom?
Well, there's something about cyanide that you guys need to know. So if you maybe look this up and bring this with you to your next lesson, we can have a little chat about it. Cool. It's an inhibitor of something important.
Okay, and so think about the type of inhibitor as well. Is it competitive or is it non-competitive? So what can affect enzyme activity and why? Again, you guys might want to pause the video and have a go at this. Tom, what do you think?
Well, so as well as obviously inhibition, there are other sort of things which can affect enzymes. I'm not quite sure what this slide is going to tell me about. Yes, so pH is an example of sort of one factor which can cause enzyme rates or the reaction rates catalysed by enzymes to change. I mean, you're probably aware of this stomach and the stomach enzymes that sort of have a low pH optimum, whereas other enzymes in your body maybe will have a higher pH optimum.
It's like protease in your stomach.
Protease in your stomach is low pH, yeah. How it works is because the pH, which is basically a measurement of the hydrogen ion concentration, can alter the charges on the active site and either, in the case of sort of pH causing a decrease in rate, it will sort of interfere with the substrate binding to the active site to form the enzyme substrate complex.
Cool. We can also have temperature that has an impact on it as well. So as you guys are well aware from last year, GCSE and everything, the more kinetic energy your molecules have, the more they collide with each other. Okay, and that's a very important factor in creating enzyme substrate complexes that lead to your products. Okay, so the more kinetic energy you have, the more collisions between your substrate and your enzyme, which means that you get more product. Okay, rather like Tom was saying earlier, you know, in order to get a reaction going, you have to add a bit of heat at first. Okay, so how can temperature affect enzymes? Again, let's have a quick look at temperature on a graph, which I just said. But if you were to plot this out, you can see that blue dotted line, that's kind of indicating the optimum temperature for your enzymes. And it kind of increases as we approach the optimum.
Yeah, so the optimum is the maximum sort of rate of reaction catalysed by the enzyme.
And so it's highest there, and then it kind of drops off fairly rapidly afterwards. Okay, so we'd like you to have a think about why that is. Okay, can you explain biologically, using the idea of enzymes smashing into substrates and colliding with them and forming enzyme substrate complexes? Can you describe that graph? Okay, and we've got another one here. So if we were to look at the effect of pH on enzymes, again you've got like an optimum in the middle, and then it sort of decreases on either side. So after what Tom said earlier on about hydrogen ions and stuff like that, can you come to your next lesson having an idea of how to explain that graph. Okay. Anyway, I think that's all from us.
Brilliant.
So thanks a lot. See you later.